Imagine knowing that one single gene mutation determines your fate. That no matter what you do, you will develop a fatal neurodegenerative disorder as you age. For many families, this isn't a hypothetical, but a before unchangeable reality. However, with advances in genetic innovation, this reality may be changing. Huntington's is a monogenetic disorder caused by a mutation in the HTT gene. Specifically, it is a trinucleotide repeat expansion, a stretch of DNA that repeats a sequence CAG too many times. These extra repeats make a toxic, abnormally long protein called Huntington that slowly kills brain cells, leading to problems with movement, thinking, and mood. The discovery of the HTT gene in 1993 is a landmark in human genomics. This allowed for researchers to discover the pattern of inheritance of the disease. Because it is autosomal dominant, every child of an affected parent has a 50% chance of inheriting it. This makes Huntington's one of the most devastating genetic disorders known. For decades, treatment only managed symptoms. Nothing could stop the disease itself. However, recently, scientists are shifting from symptom based management to gene based therapies. Using tools such as RNA interference, researchers can silence the faulty HTT gene so it produces less of the toxic protein. With CRISPR-Cas9, they are testing ways to edit the mutation directly. Both tools aim to stop the disease at its genetic source, but they work in different ways. RNA interference uses small molecules that bind to the faulty HTT gene's messenger RNA. When these small RNAs attach, they silence the message so that the toxic Huntington protein isn't produced. CRISPR, on the other hand, works more like molecular scissors. It can locate the exact section of DNA with too many CAG repeats and cuts it. The cell's natural repair system can then remove or correct the error, potentially stopping the production of the toxic protein. Together, these techniques move us from simply managing symptoms to actually editing or silencing the cause, changing the genetic script that previously guaranteed decline. Clinical trials such as AMT 130 have shown early signs of success, proving that we can target the root cause of Huntington's rather than its symptoms. AMT 130 is a gene therapy that uses RNA to silence the mutant Huntington gene, as discussed previously. Early clinical trials have shown encouraging results. In 2021, patients who received AMT 130 had a slower decline in movement, thinking, and daily functioning compared to those who received a placebo. This means that the therapy may actually slow the disease itself, not just the symptoms. These results were strong enough that the FDA granted AMT 130 Breakthrough Therapy and Regenerative Medicine Advanced Therapy designations, which could speed up its approval process. With breakthrough power comes ethical and social challenges, however. Some argue that editing the human genome is playing God and that it should not be interfered with. Others worry about unequal access. Gene therapies can be very expensive, limiting who can benefit. There are also regulatory questions. Many gene editing treatments are so new that the FDA and other agencies are still deciding how to evaluate long term safety. Innovation brings hope, but it also forces us to confront fairness, safety, and moral responsibility. It's amazing how fast genetic innovation has progressed in the past few years. The HTT gene was discovered in 1993. Almost a decade later came the first attempts to reduce the toxic protein through gene silencing in 2005. By 2017, CRISPR had entered lab trials in the United States. By 2019, AMT 130 had begun early clinical trials that eventually showed successful results. And finally, in 2025, just a few months ago, the British Broadcasting System reported the first successful gene therapy case in humans, a patient whose symptoms improved after silencing the mutant gene. In just 30 years, we've moved from identifying the mutation to potentially reversing it. That's the speed and power of modern genomics. Huntington's is more than a medical case. It's a symbol of how human understanding and innovation in genomics can change what it means to be human. What if monogenetic diseases like this become curable, or even preventable? What if the next generation never has to fear a family inheritance of suffering? For the first time, our DNA is not an unchangeable blueprint, but a canvas for changing and treatment. If genetic innovation continues at this amazing and unprecedented pace, we may be closer to a cure for Huntington's than many people think.